Assalamualaikum warahmatullahi wabarakatuh. I am Nur alias SAFIYA binti MOHAMMAD HAZUAN, metric number 82115552. Today I will present my scientific poster entitled Lichen Biomonitoring of Airborne Microplastics, a comparison of urban, natural and protected environments. This poster is based on the original article by Torozzi, Galitelli, Cesarini, Romano, Orsini and Scalisi, published in 2024. The authors are affiliated with University of Roma Tre, CNRIRSA, Roma Tre University, and the National Biodiversity Future Center in Italy. The study evaluated epiphytic lichens as low-cost passive biomonitors of airborne microplastics across 3 sites in central Italy. The study also validated a hydrogen peroxide digestion method and found increasing microplastic accumulation from the natural site to the urban site, including the first evidence of airborne mesoplastics entrapped by lichens. As shown in the map, the study sites are located in the Latium region of central Italy. The 3 sites represent different environments, which are the natural site, protected site, and urban site. This helps the researchers compare microplastic accumulation across different levels of human activity. For the introduction, airborne microplastic pollution remains largely under-investigated compared with pollution in marine, freshwater, and soil environments. Atmospheric fallout is an important source of microplastics, and more urbanized sites generally experience higher deposition. Lichens are excellent biomonitors because they have a high surface area to volume ratio, simple anatomy and no root system. They can physically capture airborne materials and provide a cost-effective alternative to instrumental monitoring. However, their use for detecting airborne microplastics remains poorly investigated. The first objective of this study is to investigate the bioaccumulation of airborne microplastics by lichens along an anthropogenic pollution gradient. The second objective is to compare microplastic entrapment among natural, protected, and urban environments. The third objective is to evaluate the effectiveness of lichens such as Cladonia and Xanthoria as passive biomonitors of urban microplastic deposition. For the research methodology, a total of 90 lichens were collected from natural, protected, and urban sites. At each site, 15 Cladonia and 15 Xanthoria samples were collected. For the analysis, samples were externally observed, digested using 30% hydrogen peroxide, vacuum filtered, and examined for entrapped plastics. Polymer types were identified using micro-FDIR, followed by recovery experiments and statistical analysis. The method validation showed 100% particle number recovery and 97% mean weight recovery. Moving to the results, 253 plastic particles were detected across 90 lichen samples. The natural site recorded 58 particles, while the urban site recorded 116 particles, which is about 2 times higher. This shows that microplastic abundance increased from the natural site to the urban site. For the particle characteristics, fibers were the dominant form of airborne plastic detected in the lichen samples. Fibers made up 97% of the particles, while fragments made up only 3%. For fiber length differed significantly among sites. The natural site showed the shortest average fibers, while Rome, the urban site, showed the longest average fibers. For lichen genera, Cladonia entrapped 108 particles, while Xanthoria entrapped 145 particles. However, their accumulation efficiency did not differ significantly. This means both genera can be useful as passive biomonitors. For polymer identification, cellophane and PET were the dominant polymers. PU occurred in the protected site, while PAN occurred in the urban site. In the discussion, microplastic accumulation increased from the natural site to the urban site. This suggests that urbanization and nearby human activities influence airborne plastic deposition. The high accumulation at the protected site may reflect wind transport from nearby urban areas, particularly Rome. The dominance of fibers may be related to their low weight, which facilitates atmospheric transport and entrapment on lichen surfaces. This supports the idea that lichens are effective in trapping airborne microplastics. In conclusion, lichens effectively trap airborne microplastics across natural, protected, and urban environments. Their wide distribution and low monitoring costs support their use as passive biomonitors of airborne plastic pollution. However, further studies involving more lichen species and seasonal conditions are needed. For the acknowledgement, the original authors thank the funders, students, research site management, and reviewers who supported the study. Overall, this study shows that airborne microplastics are an important environmental issue, and lichens can be used as a low-cost and effective method for monitoring airborne plastic pollution. Thank you.